So okay, let's start. So first of all, I want to introduce myself. So probably some of you already met me before because I was like teaching some of you about the beginning and introduction to the Python programming. And now we will switch and we will learn about bioimage analysis. So I am currently working as a computational biologist in WashU. And so my work somewhat related to image analysis from time to time. And we will talk about it from the angle of biology, immunology. So the idea that we will take a look mostly on the bioimages. And when I say about bioimages, I mean like, so for example, image of the nuclei staining or like image of immunofluorescence where you have different antibodies captured and acquired by some technologies. And we will mostly be working through this course. So yeah, basically, I have the website which will be uploaded meanwhile we are working. So right now it's like work in progress, but I'll share the link to it with you. So and you can go over it with me. So during the lessons. And yeah, so the requirements for the course, I already posted it in the Telegram channel, but I want like to make it clear. So for me, it's very important that you're like joining the lessons because so it's an online course. And for me, it's important that you are like really present in the class and also that you are using the web camera because otherwise for me, it's like very hard to get any feedback and it's like required. So in case if you want like to finish the course, you need to do it. There is no like other alternative option. And also, so closer to the end of the course, I will provide each of you the mini project. So the mini project will be in the following way. So basically your task is like to take a look on some particular publicly available data set with the spatial proteomics and analyze it. So this is the main core idea. And on the website, basically, there are some sections which we will go over. And in this course, we will try to learn about bioimage analysis. We will try to work with different type of imaging. And the idea mostly in this course that we will cover one particular task in bioimaging, which is cell segmentation. So it's like very kind of modern field. So there are some like solutions which are already, for example, surpass the human precision in terms of the annotation of particular things. And in the cell segmentation, what is the task is like, so for example, you have the image and the idea is like to identify particular individual cells. Here are just like some nice examples that I found in terms of the how they look like. The idea is like to identify particular cells and assign a mask to the cell. So in this course, we will try to get to this point where we can write our own like simple, for example, script that can take the image, perform cell segmentation, and measure different features for us. And this is a sum like of the kind of what's will be in the course. You can try to play around and right away execute code. So basically on the website there are some code snippets that you can copy and paste in your editor, and basically they're supposed to work. If they don't work, please like let me know, because as I said, it's like work in progress and there might be some issues with the implementation. But basically the idea is that so we will work with analysis of the images with inside of the Python. And I hope that most of you already know some basics in Python and you can understand like main ideas behind the code. So and there are some prerequisites. So as I said, you need to know Python programming language, and it's like very nice if you know NumPy and SciPy, like some basics of it. And also that you should be able to work in command line interface, because in my mind, like bioinformatics is like heavily based on the different tools which are like inside of command line interface, and we will from time to time probably will work in the terminals and do it there. So yeah, hold on. Okay. Okay, so and let's start. If you have any questions, so like during the course, please ask your questions. It's like I'm very happy when people like asking something. I'm not sure that I will be like 100% times like. So I mean, like, it's not guaranteed that I always know the answer, but at least I will try to help you with some of your questions. Yeah, and basically, so you might ask, like, why should we even bother ourselves with the learning of the bioimage analysis? It's like some, so maybe for some of you it would be as some orthogonal field. You're like, okay, I want like to perform data analysis of single cell data, or like, for example, I don't know, something that is related more like in to you in terms of the bioinformatics. But the idea is that right now the field in the area where it's like trying to push forward in the way that they're trying to build new techniques and new methods to identify both modalities. For example, if you already saw the single cell data analysis, please let me know if you already saw some examples of the single cell data in your course before. Did you see it something like that? No? Okay, basically, but so for example, did you work with Fantasus, right? So you probably, all of you saw Fantasus as a tool for visualization of the bulk RNA-seq data. So in bulk RNA-seq data, the idea is that you usually mesh all the cells that you have, and then you profile and measure some like average profile of the gene expression levels per gene. In single cell, it's like somewhat similar, but the idea is that your final output is a matrix where you have a huge matrix with features. Features are genes, and columns usually, I mean, you can think in it about it in different ways, but you have genes and cells. For each cell, you have the gene expression level. And now, so basically, if you take a look, there are some links to the nature methods. This is like one of the high impact journals which are publishing some methods related to some, I mean, to the, for example, bioinformatics. And from time to time they have a release where they saying that there is a like a modern method that will probably change the field. And for example, in 2020 and 2024, they said that spatial proteomics and spatial transcriptomics are the cutting edge technologies. And basically, so for example, spatial proteomics is built on top of the images. The idea is that you have images, and then you need to analyze them to get some of the understanding behind the biology and like to be able to answer to some questions related to biology. So, and you can take a look, read about that, and then probably it will kind of make you more confident in the way that you need to know some of this to be able to derive some hypothesis or some ideas when you analyze images. And in this case, we will work in Python, and basically the first step that we need to accomplish is like to create an environment for us. In my case, I usually use Conda or VNF. If you prefer something else, there are like a lot of different package managers or something like that that you can use in your case. It's okay, so it's up to you. In my case, so I'll follow the instruction which is like for the Conda. So let's create a Conda environment. In my case, I am using the VSL2, which is a like Linux-like system, but inside of the Windows, and you can do the same. So let's create an environment, and then let's install some of the packages that we will be using in our course. So, and you supposed to follow, because I mean, it's nice to just like to look and hear something, but you get knowledge when you try to do it yourself, and then when you face some problems, you get experience. And in my mind, so you need to try to follow it. So okay, in my case, I created the environment. Now I can activate it by using the conda activate bioimage in my case. And let's install all the packages that are provided in the instruction. So I'll just briefly explain. So there is like, for example, OpenCV Python. It's a library for computer vision analysis. NumPy is for numerical Python. Matplotlib we will use mostly for data visualization, and scikit-image, it's a package somewhat similar to scikit-learn, but there are some methods which more related to the image analysis. Along the course, probably we will install more of the packages. So, but for now, right now, we are good with this list of the packages. So it should be enough for today's lesson. So guys, so far so good. Can you create an environment? Is it okay? If someone, so guys, if someone like have any question, please let me know, because we
Getting
want.
package metadata.
What, sorry?
It's getting package metadata, and it's typically not very fast with content.
Okay, yeah, fair enough. You can use, as I said, so for example, I don't know, I heard that there is like, for example, Pixie as a package manager or UV, so whatever you want. But this is like mostly more common and more like popular, so should be good enough for everyone. In my case, so I activated, installed everything, and now I'm ready to go. Let's create a folder. So in my case, I called it like Bioimage 2026, and I'll create a new folder. I will call it Vic001, something like that. I will open it in the Visual Studio Code, and then we can start to work with the images. And before we start, so I want just like to highlight that probably all of you already know some type of the images. So for example, when you are working, so for example, if you want to post some image to Instagram, like this like forbidden social network, right? If you want to post it there, usually you have on your phone, for example, image in the format of JPEG or PNG, something like that. It's like common formats. And in case of the biology, bioimaging, we usually have some particular data formats. And the idea is that beside the image itself, we also have some metadata. So for us, it's usually important because in regular images, for example, taken on the phone or on the camera, there is also some metadata included to the image, but it's not enough in terms of the biology. In case of the microscopy or something like that, we usually want to know, for example, the size of the each pixel, because the idea is that when you do microscopy imaging, you should know what is the size of the each pixel, and then you can reconstruct, for example, in terms of the size, how large was the cell or some other features. For example, you can also store inside of the microscopy image in terms of the metadata. You can have the exposure times for different channels that you had, and it's very important. But beside that, the idea is usually the same. So the core or the backbone of the image, it's like a numerical array. Because each image, we can think about it in the way that it's a numerical array, and then it's much easier for us to understand what's going to happen. So here we have an example. This is so one example that I took from the internet of the like a nuclei staining. So on the left, you have the image of the nuclei. You can see particular nuclei of the cell. And if we zoom in to particular area, like very close zoom in, you can find, I mean, like when we open the image inside of the Python, we will find out that this is just numbers. And how it's done, basically, you have the numerical array, two-dimensional, for example, in case if you have a grayscale image. Grayscale image, it's a black and white image. And in this case, we have a numerical array with the height and width, basically two dimensions, and then inside of each cell, or like each entry, it's a value. And in this case, you can see that the dim areas of our image has values which are like closer to 0. So here they're not indeed 0, but they're like closer to 0. And in case if it's some bright area, the values are going upward. And usually, so there are different formats how to store the data. In this case, the pixel value can fluctuate from 0 to 255. It's the idea if you have like this type of the image, you will have, for example, the range of from 0 to 255. If you have a more like a higher resolution or like high quality image, the range could be bigger. So for example, from 0 to 65,000. I mean, there are some values in the trailing because it's like always the power of 2, and basically it like represents how many bits of information you can store in particular pixel. And so the idea for us like that we need to keep in our mind that this is just a numerical array, and then you can right away start thinking about it in the way that what kind of methods that I already know I can apply to the image. So let's try to brainstorm a little bit. So what can you do, for example, with the image if you think about it as a numerical array? It's like just kind of you can provide any type of ideas that you have. You don't need like to be thinking in the way of bioimage analysis. It's like just creative idea that you can have. Guys, don't be shy, please. Let's try to communicate. Any idea?
Maybe try to count the sum of columns or rows.
Yeah, for example. Something like that. And then we can, for example, identify which row has the highest value or something like that. Yeah, what are other ideas, guys, that you have?
For example, we can bleach it and find details that are not visible in the color version.
Okay.
We can take some sigmoid function looking graph to increase contrast.
For example, yeah. But basically, yeah, so the idea is that you can use whatever you want, and then the beauty of the images usually that, for example, any operation that you have in your mind, you can right away test it against your image. So for example, you can say, I want to, I don't know, find the mean value of the image. Since it's a numerical array, like, okay, there are like h multiplied by w values because we have height and width. We can identify mean value, and then you can say, okay, I want like to say if the value is higher than mean, let's say that it's true, and if it's lower, then it's false. And then you can project it to the data, take a look, and you will see that you converted the original image into the binary format where you have only 0 and 1s, and then you have the representation which areas of your image are, for example, higher than the average. Yeah, so this is a representation of a grayscale image where you have black and white. And any type of the colored image, so for example, if you have a color image, the idea is that usually you have the extra dimension. So you have stack of 3 images. Each one you can consider it as a grayscale, but you have 3 of them. And basically the first image in this stack represents the red color, the second is the green, and the third one is the blue. So, and this is a representation. Basically, if you have just like grayscale image, it's one single array. And then in case of colored image, we have usually red, green, blue stack. And then basically the combination of red, green, and blue provide us the different colors that we can see on the, for example, on the screen or on the monitor. And in the bioimages, for example, when we will work with the spatial proteomics data, we will see that there are like even more channels. It's not just 3 channels, it can be like 20, 30, 50 or something like that. And the idea is that we will mostly be working in the beginning with a single channel. and then we will learn how to apply it, for example, to the colored image, or to apply it sequentially for each color array that we have. And let's start. So we have the first code. I will just create a new file, and I'll call it so like a lesson.ipynb. So okay, I want to create a file. So in my case, since I am working as in Jupyter notebook style, it will ask me to install one particular package in the beginning, but that's okay. So let's just copy paste the code that we had. I'll try to execute it. So it will ask me which kernel of Python I want to use. In my case, it's a Bioimage, because I installed the Bioimage, and this environment contains all these packages that I'm going to use. So now I'm trying to run it. One more time, it asks me that I need to install ipykernel package to be able to work in the way of IPython notebooks. So it takes some time, but then in the end, so we will be able to execute it. While it's installing, I want just to go over the code that we created. So because the idea is that we want to understand what is happening. So right now in the era of LLMs and all of this, they can spill out a lot of code to you, and for us the best way to learn something is to understand what is happening, not just to blindly execute code and be happy with that. We want to understand what is undercore or underneath. In this case, so we exported from the package which is a scikit-image. We imported IO. It's for input-output utils. And then we also imported matplotlib pyplot as PLT. So this one, I hope that you already used it before, because it's like a general package for the visualization of your data. And then I created a URL variable. So this is basically URL which leads us to the GitHub repository. And basically this image stored in the GitHub, and we can access it. So as you can see, I opened it in the web browser. This is the same image that we saw in the example before. This is a nuclei staining of some sample. And yeah, so now as you can see, we specified the URL, and the nice part about IO module from the scikit-image is that we can read the image directly by using the URL. So this image stored somewhere in the web, but we can provide the URL and it will automatically do the job. So it will create the image representation inside of the Python. In your case, so what can you do? And probably it's better in my mind, because right now, so this image is it doesn't belong to you, somewhere in the internet. And for example, if there is some problem with internet, you will not be able to open and execute the code in the same way. So in this case, you can, for example, go and download the image itself. You can, so for example, save image as and store it in the same folder where you have the notebook, for example, and then provide not a URL, but the path to the image, and it should work in the same way. So you can be able to run, for example, in case if you have a path, you can write, so for example, if it's relative to your current directory, you can specify that you have depi png if you save the image and called it as a depi png inside of the same folder. And here we just then later, using the PLT functionality, we use image show. Basically, it stands for image show. We use CMap gray, and then this is just, if be honest, this can be excluded. I just added it to have better visualizations in terms of how it looks like on the website. But this part is not very crucial for us. So for example, you can avoid it, you can remove it, and in this case, you have the axis for X and Y, and also the background is white. But the idea is basically the same. So we just represented our image. And then you can try to play around, for example, to change something in your code to get the better understanding what is happening. So for example, in this case, we have CMap gray. Let's remove this part and just run the program without image CMap gray. And in this case, you can see that the Python by default, when you use image show with a grayscale image, it uses another like lookup table. So, but by lookup table, I mean, so as you saw before, we had values inside of our array, right? And in this case, the color map that we assign to each value is like starts from black to white, where black corresponds to 0 and white corresponds to 255. And in this case, we can like kind of visualize the data using the gray color map. But by default, it's something else. And here we just simply changed it to gray. Okay, so let's return it back. And now the idea is that, as I said, this is a numerical array, and we can use the same functions as we used before, or maybe we will use them first time today. It's not a problem to understand some features of the numerical array that we have. So in this case, I will copy paste this part of the code. Let's execute it. And as you can see, we have the first, it's a shape. Basically, image shape shows us what is the size of our image. So in this case, you can even look to the image that we created with the PLT module. In this case, you can see so that the height is slightly larger than the width. In our case, width is almost 500 and the height is like 500 something. And then when you take a look on the image shape, you see that it's like 520 by 502. And basically this is a size of our array. Then we can check the type of the image. And as you can see, it's a numerical Python ND array. And the D type, it's unassigned int 8. And also we can check min and max values inside of our image. So is it like okay-ish? Are we on the same page, guys?
Yes.
Okay, so then let's try to execute the following task. So the idea is that we want like to, as I said, this is a just array. So in case if we, for example, run it as an image without any function, you can see, so this is an array, like 2D array. Let's solve the following task. So we want like to measure the average or mean. Calculate average, and then I will not put it here because otherwise it's gonna automatically autocomplete it. So the idea is that we want like to calculate the average value of the image and then create a new image. Let's call it like bin image, like binary image. And we want like to assign image which is it's like very hard with the autocomplete. Basically, so yeah, the idea is that we want like to create a binary image, and for example, if the value of the original image was higher than the average, we will assign 1, and otherwise we will assign 0. So let's try to execute it, and then the idea is that you need to use PLT show to display the image. So you have time, please work on it. And if you finished, please raise your hand so I will be able to understand how many people already resolved the task. Guys, while you're working, so can you just give me an idea? Did you have already somewhere like some examples of NumPy? Yep.
Yes.
Yeah, okay, but are there like people who didn't see it before? So as I understand right now, so we have like the full group, right? Not like a splitted by the like programming or biology. So for example, guys who are like from the biology specialization, have you already seen in some other courses like numerical Python or something like that?
Yes.
Yes? Okay, great. So
Yes.
then basically you should know all this, like that you can like calculate the average by using some functions which are built in, or if you want, you can like implement your own. And then there is like some masks which you can use inside of the Python.
Do we need to calculate the average like per row, or just as a flattened array, like the default NumPy min?
So we want to calculate average of the whole 2D array.
Mm-hmm.
Yeah, and there is a function. So, okay, I see that some people finished it. Yeah. Okay, nice. So, in this case, let's do it in this way. So, can someone like just put it to the Telegram channel? And we will check it. Yeah, but we want like to generate an image. So we don't want just to calculate the mean. We want like to generate a binary image where instead of the values from 0 to 255, we want to assign, for example, if the value inside of the, okay, I can put it. So the task is to, okay, so there is a one possible solution. I'll just put it here. So this is an example of how it's done. Yeah, let's import NumPy as NP. I'll rerun this part. So, okay, and here we have the solution, right? And let's then use plot im show by image cmap gray. So, and let's execute it. So here we got the result. So, Ivan, can you tell us just like, it should be simple, but can you just explain what's happening here?
And every value in the image that below the average 0 value and above 255 value. That's all.
Okay. So guys, any comments about this part? I mean, about the solution and maybe in general. Do you have any concerns maybe? But looks interesting, right? So let's just a little bit describe the output that we got. So as you can see, now we have like a binary image. And basically, if you check, for example, image unique, okay, if you use np unique of the image, you will get that there are like just two unique values, 0 and 255. In general, so you could do it even like a faster, but the idea is the same. So now we have like just two values, 0 to 255. And then you can see that somewhat we already got, so basically we solved one of the tasks. It's like one of the possible approaches how to resolve the task, which is called image thresholding, where we want to separate, for example, background from the foreground. In our case, it's not the ideal solution. So you can see there are, for example, this part is kind of on the edge, and probably we would like to have it as a foreground because it's not indeed like background. But this is one of the possible ways how we can do it even in this small and simple example. I would probably like a little bit discuss it because so in the image analysis, what we want to do usually, it's like to always keep in mind that there is a original image that we had before. And usually we don't want to change the original image in our program. So we want like to, for example, have a workflow, a pipeline that applies different like tools, methods to our image, but the original image we always want like to store somewhere. In this case, we don't like really worry about that because we want that the original image stored somewhere. So it's like stored, for example, as a local image in your folder, or it's stored in the URL. But in this particular case, for example, right away, I already don't have the access to the values that they were before. And in case if I want like to get again the values, I need like to reread the image. And in the analysis, it's always like as a kind of as a balance. You usually think about the memory that you're using in your program. So for example, sometimes images could be like very big, and in this case, you don't want like to create new objects in memory and to overflow your memory. And in this case, okay, you would like to, for example, load the image and then apply all functions right away to the object. But in this case, since the image is like small, I would probably do it slightly in another way. So I would like, for example, rerun this part again. So now image it's again not a binary image, but image of different values. And I would do it in this way. So I would create, for example, the binary image. I would call it like binary image. And in numerical Python array, the beauty is that you can right away use image is greater than average. Let's remove this part, and then we have binary image. So let's just substitute image by binary image. So the result is like identical. In case if we execute like bind image, we will get that there are two values, false and true. Now we kinda also not using the NP unsigned int 8 anymore. It's a boolean, so probably in some cases it could be better in terms of the memory efficiency. But in case if we check, for example, the original image, we will see that there are plenty of different values inside of it. So we at the same time have our original image, a new one, and in case if you want like to directly adjust original image in any way, we still can do it. And this is just really important to keep in mind because, for example, the big images of spatial proteomics, they could be easily like, for example, 60 gigabytes in case if you try to load them to the RAM. And in case if you try to load them to RAM, if you're working on your, like, for example, laptop, you'll face with a problem with the memory overflow. So if there is a raised hand, so is it like raised because of the question or... Artyom, sorry, do you want to say something? No? Okay. So yeah, now let's go further. And now our task is like to implement very simple function. So when we're working with images, we already want like to, mostly all the time we want to know the, for example, the data type of the image, shape, how many dimensions inside of the image, and let's execute it in the following way. So this is just as a description of what we want to execute, and this is the implementation. So we will define a function which is called inspect image, and let's just use it in our program. So here I have the new function, and let's just inspect our image that we had before. Yeah, so this is what we want to execute, and this is the name that we assigned. And then basically we go through different attributes of our image array. So first of all, we use type of image, and we got that this is a ND array. Then the shape, it's 520 by 502. D type, it's unsigned int, so we have minimum value, maximum value, and inside of the dimensions we have just how many dimensions we have. In our case, we have height and width, and then that we think that this is a grayscale image because we have just one particular channel inside of the image. So guys, any comments to this part? Okay, let's try to create now, for example, colored image, so image with colors. And in this case, you can do the following thing. So for example, you can find the image in internet, which is color. In my case, I will just use np random seed, for example, 42. And then let's create a random image, np random. So as you can see, it's like helps me to create some image. In case of the size, I want like to have a color image, and I provided the third dimension 3 because we want to have red channel, green channel, blue channel. And the type is unsigned int. So in our case, we will have image like 100 by 100, and then we can plot and show the image. So let's execute it, and as you can see, we got some random colored image. And by default, if you have the red, green, blue channels defined, or if you have 3 channels inside of the image, Python and matplotlib will think that you have red, green, blue, and it will use proper representation of the image. So, and as you can see, I just created a numerical array, random values there, from 0 to 255, and then you can visualize it. So, and again, I just like want to probably kind of keep focusing on it. In my mind, the real beauty of the working with images that you can right away see what's happening. Because sometimes, for example, if you analyze something which is like numerically represented and you don't know what's happening. So for example, you can work, I don't know, let's say with gene expression data, you are like having like one sample and then you have gene expressions. Okay, you applied some of the like normalization tools or something like that. And then, okay, you can check like histogram or something like that, but you don't see, for example, how it's like structurally changes the data which is underneath. Here it's like right away, you can like use plot im show and check what was before and what you got after. And in this case, it's maybe easier to, for example, learn some techniques. You can read about them and then try to apply them to the images, and you will get the right away the representation of what happened after your tools were applied to the image. So, and now let's check particular images, not particular images, but particular channels in Python. So in this case, we will use a data example that is provided by scikit-image package. So there are some images which are like just stored inside of the package, and we can use them in our program. So in our case, we will use scikit-image data and we will import color wheel. Then we will again use matplotlib plt to plot the image. So let's just run it. And as you can see, we got the color wheel in our output. And as you can see, so we have the shape of the image again. Like we have 3 channels and the height is 370 by 371 the width. And now we can access directly particular channels in the image. So for that, we will use the following code. So if I go too fast, let me know. So we will do the following thing right now. So we will plot a figure. Basically, it's a canvas where we will locate our images or like our charts later. So in this case, we using the subplot function, and we are saying that okay, we want like to have one row with 3 columns, and the first entry is going to be image, and here we get a slice. So guys, do you remember what a slice in Python? Nice, that's cool. So yeah, we got the slice and we get the first channel in our image, because for the first dimension we use all the values as a slice. For the second we use again all values for slicing, and here we just saying that okay, return us only the first channel. And then you can see that we use particular CMap for different calls. In first one we use reds, greens, blues. So let's just execute it, and now we got the output. So as you can see, the channel 0, if we use the CMap red, looks like this, then green, blue. And let's just take a look on the particular call, because we know in advance that this is basically the typical structure of the data. Usually when we have colored image, people store it in this particular order: red, green, blue. But order can be different, and for example, other packages could work in a different way. So for example, there is the OpenCV package, and when it reads the image, the order of the colors, it's not red, green, blue. It's blue, green, red. Another order, and we will see the example right now. So and in this case, we know that in scikit-image, the images stored in the way that the red is first, then green, then blue. And because of that, we can assign manually the CMap here. But if be honest, for us, like generally, it's not very important, because any image is just an array. And in case if you use just no CMap or default CMap, it will be looking like that. So CMap, it's basically how you assign the color map to values that you have inside of your array. In this case, we assign like red to be 255 and 0 is like white. Okay? Guys, is it clear about the order of the channels? Okay. Okay, then let's take a look on the following example. Yeah, so let's try to execute, let's solve the task first, and then we will take a look on the OpenCV example. So right now our task is like, we have the image and we want to create a copy of a original image, and green channel is removed. I mean, by removed, it's like still there, but we want like to make it 0. Yeah, and red, blue still the same, unchanged, and then we will get a magenta representation of original image. So you have some time, guys. You can use the same example. So for example, color wheel, you can load it as it's like described here. And then the idea is that we want like to get rid of the green channel. Yeah, and in the same way, so guys, if someone has resolved the task, please raise your hand, so then I can understand that someone resolved it, and then we will discuss later steps. Yeah, and while you're working, so I also want to point out one idea. So basically, when I was working on the course, I was like asking myself, so okay, I want like to tell you about some classical computer vision methods that still exist and still broadly applied. But for example, you might ask, why should we do that, right? So for example, you may be already using all these generative AI techniques to generate images, or like to use them to adjust images. So guys, how many of you, for example, takes images on the phone and then using, for example, Lightroom or I don't know, some editor to get rid of the people there, for example? Does anybody do that?
Me, I use Photoshop and Adobe Lightroom.
So yeah, and then probably you saw, right, that there is like generative AI. You can just like draw a mask and say, okay, I want like to get rid of it, right? Okay, I kind of assume that yes. And basically, so there are like some other techniques which are like built on the generative AI, but the problem is that you change the image and you don't know what exactly happened. But in case if we want like to do some bioimaging analysis, for us it's very important to be able to interpret what happened from one step to another one. So we will also adjust images, we will change something in them. But the key point is like that we want like to see what was before, what was after, and then also like to be able to say, okay, is it fine enough, or did we over computed something or over processed something? Because we will see that there are some cases where we, for example, lost, or we are like, by doing some steps, we are losing important information. And with generative AI, sometimes it's like tricky. There are definitely some neural network based techniques which are like people using, and we will see it indeed. But we always should be cautious about that. So, okay, Artem, can you share your solution in the Telegram channel and then can explain it? I'll just put it on my screen, but that you can explain what you did. So, okay, here we got a solution.
So yeah, we just made all the green intensities 0 and got the magenta.
Nice. Let's execute it. Yeah, but so besides that, everything the same. So the shape of the image is gonna be the same. we just changed the green channel, which is like second in order. Okay, and if we do it, so you can see this is a magenta kind of representation. We don't have greens inside of the image. And it was like kind of pretty straightforward. We took the image, we accessed the second channel, which is green, and we just like wiped it off. Okay. So now let's take a look on the example with CV2, which is OpenCV package in Python. So I'll copy paste this example, but before that I want to save the image first. So let's repeat this part. So I'll read the image color wheel again before executing this part, and I want to save the image color wheel PNG. So let's execute it. So we save the color wheel as a PNG file on our PC. So as you can see, this is a color wheel PNG in the same folder. Probably I can like slightly increase it. And then we can do the following stuff. So we can now so we can read this image by using the OpenCV package. So let's provide the name of the object. And as you can see, so right away the Visual Studio Code, because it's like with AI autocomplete, knows that something I'm doing wrong. It's like, okay, probably you want to use CV2, convert color from image CV, blah, blah, blah. But I'll avoid it. So let's just execute it. And as you can see, so the red was on top. In the first run, red was on top. And now red is on the left. So something changed. And basically this is a key point about that different packages might work different, and we want like to know about the description of the methods, what they expect and what they will output. So there is a function which is called cv2.convertcolor from blue, green, red, because by default when we read it with OpenCV, it's like blue, green, red to RGB. And then if we do that, we can use again plotImShow, because plotImShow expect that the first channel is red, second green, blue. Okay, but let's try to solve this task without using the CV2 convert color, without this one. So our task is like to read the image by using the OpenCV package, and don't use CV2 convert color and get it from the BGR to RGB. So in the same way, so if you solved it, please raise your hand and we will take a look. So guys, let's try to think about it together then. I see that Ivan, you solved it, but let's try to just discuss. So Ivan, can you just maybe as an idea, not just as a solution, but what you used, for example, as a method to change the order, what you saw it in your head before?
I rearranged the color column, like just reverse it, because RGB and BGR, it's like reversed.
Okay, so sounds good. So you want to notice that RGB and BGR, it's like kind of similar, but the order is different. So it's like inversed one. And do you guys remember, for example, in terms of the slicing, how can we read, for example, if it's like just one numerical array, just one dimensional array, how can we read it from like back to beginning?
Skip -1.
Yep. So and now we can try to use it inside of the in our program to reorder the original image, because numerical Python, so it like uses the same ideas of slicing behind that. It's like just one of the possible solutions. So for example, you can create it You can definitely use a lot of different things, but this one probably like the shortest one that can be used. So okay, let's try to spend 2 more minutes, guys, and yeah, please let me know again. And I hope that there are no people who solved it and don't raise the hands, because don't be shy, so everything fine. It's a friendly environment, right? We're just trying to learn some stuff, not to bully each other. I hope so. Okay, or let's do opposite. So if you want to bully someone, bully me, but not your classmates. I'm ready to take the fire. So don't worry. If you, or if, for example, if you want just like to discuss, you also can raise the hand. You're like, okay, I don't know how to solve the task. Here are my ideas. Let's just try to kind of talk about them together. So that's also fine. Okay, Dastan. So can you share it in the Telegram and explain it? Okay, here we got the solution. I'll just put it here. So, okay, this is a image that we read from the scikit, the image that we got from the OpenCV. Yeah, but where do we change the order?
One second.
Yeah.
I thought that this part is already over.
Okay, so here we got. So in this case, we probably want to use OpenCV, right? So yeah, can you just briefly go over it and explain? I mean, probably we don't want to have all of this, so this one we can comment and just focus on this part.
So it's, as we have already discussed, so if color channels just need to be inverted in order, you can use slicing, and skipping 2 parts means just we iterate through entire list without skipping anything. And -1 means that we just listing it in the order from the end to the beginning.
Yep, right. So, guys, any questions about that? No? Okay, no questions. But I just want to get a grasp of ideas that you had. So, for example, guys who tried to solve it in another way and didn't have enough time to solve, no, I mean, like to accomplish it. What was your idea? Do you have any things that you can share with us?
I tried to make a variable corresponding to each color and reset them in the right order, but the image came out grayscale, so I think it's a wrong solution.
I see. Let's try to do it together, guys. So I'm not sure. So as I got it, you want like to get the red channel, right? But
Yeah.
let's use image. Yeah, in my case, the Visual Studio Code gonna suppose to help me. Yeah, so here we got the RGB, but yeah, in case of the OpenCV, we know that they are like in the another order, right? So something like that. We know that R is the last one, green is the second, and blue. And now we can create a new image. So image RGB is NP stack. So yeah, basically it helps us to stack the channels in proper order. But basically, so here we just provide RGB, and let's check whether the Visual Studio Code is smart enough. Okay, I did not execute this part. So yeah, as we can see, so red is not where it's supposed to be. And now, oh shoot, hold on, what's happening? Yeah, why it's...
Like everything right, and you have a right image now? Like everything rearranged?
Ah, yeah, right. So I'm looking on the previous one, which was like reordered. Yeah, you're right. So because this is the one that we read with OpenCV, right? So this one that we read with OpenCV. And probably the best way, like, not to get like tricked again. So this is to use the following stuff. So okay, we created the image RGB, and we want like to check before after. So let's do it in the following way. So I will create figure axis, plot, subplots. I want to have one row, two figures, figure size. It's okay to have something like that. And let's just use these two things. Yeah, so something like that. And then basically, so this is the left one that we read using the OpenCV, and then we reordered it by using the this code. Yeah. Yeah, so and as you can see, so the Dastan sent another way, which is like also, for example, basically does the same thing, just splits the image by using the split function, and then we use the merge from the OpenCV. But in my case, usually I try to use NumPy because it's like easier for me to kind of, because in this case I need to know just, for example, some methods from NumPy array, and then I can just apply them. Okay, so let's go to the next thing now, and this is about some image intensity and bit depth. So we can face different type of the data along the course, and so for example, the common one in my mind, it's like 8-bit image when we have the values in the range from 0 to 255. But sometimes, for example, if you do some microscopy images, for example, on Leica microscope, you can get as a raw image 16-bit image. It's like more information stored in the data, and sometimes you can see that, for example, if you do the compression from 16-bit image to 8-bit image, you can lose the important signal from the image. And this can be sometimes tricky, that you need to keep it in mind that the depth of the information inside of the image is important. Then also there is a float representation of the images. In this case, you have that the value are in the range from 0 to 1, I mean like as a float number. But basically, do you have any like thoughts about it? For example, what are the advantages and disadvantages of these types? Something that comes to your mind.
Resolution, occupied space.
Yeah, occupied space. So this is like very important because, for example, if you have like a large image, it's very hard to store it as a float image because it takes much more space on your storage rather than, for example, 8-bit. And because of that, some some companies might prefer to store their images in an 8-bit depth. But it's like you need to be kind of cautious about that, and remember that sometimes if you have, for example, the weak signal for particular antibody, it can be kind of lost in the final image in case if you, for example, convert it from the 16-bit to 8-bit. Not all the time, but in rare cases it could happen. Yeah, and so now we can, for example, take the image that we have. Let's say that we have image. We can make a flattened version of it, just like as a 1D array, and represent it as a histogram. So in this case, we see something like that. So let's just copy that and paste. Yeah. So, and in our case, we want to read images io imread URL, the same image that we had before. So let's execute it. And as you can see, so this is a representation of our data. And basically by looking on the histogram, we can get some idea of the image. So for example, guys, when you look on this histogram, do you have any kind of thoughts that you have in your mind? So for example, you didn't see the image first and you just got the histogram with this like nuclei staining, and now the idea is that whether we can like to say something about the image even without looking at it.
I think this image is pretty black and it has so small white values and it has a lot of gray values like
Okay.
that, black and gray.
Yeah, correct. So you're saying that, for example, we don't have like a lot of values on the right side of the histogram, right? And mostly they are concentrated somewhat in the left side of it. And there are a lot of dark parts in the image. So maybe some other guys, some other ideas. So guys, are there like some data scientists?
There are low contrast between in picture. I mean, a lot of gray. Number of pixels in gray very high.
Okay. Maybe some
Full
other.
model distribution.
Yep, right. So we have like two part of distribution, right? So for example, you can think about it that you have like a distribution on the left, which is like more darkish, and then you have the second one. So this is a data scientist response. He's like, okay, I see two distributions. And basically this is sometimes important because in some cases you want to have two distributions to be able to nicely separate, for example, one kind of structure of the image and the another one. Yeah, and in this case, so we have somewhere the threshold between two distributions. Yeah, so and also sometimes you can see, so in case if you look on the example here, so you can see that there are like a lot of values around 0 and a lot of values around 250. And sometimes, for example, we don't want to have it in the image. So it's better to have the image where the values are not touching the extreme points, because that means that the information there is lost. So for example, if you do photography, you might know that there are overexposed areas or underexposed areas where you don't have enough of the information and you cannot understand what is happening there. Because for example, if your original image, there are a lot of values which are like on the edge of 255, you cannot say, is there like any difference inside of this population of the pixels? So, and this is just like that you can think about the image in terms of the histogram. Yeah, and also let's finish today with this part. So I will very briefly go over. So that the problem that we always have in the imaging, it's a noise, which comes from different type of sources. And usually you want and I want to have an image with a high signal to noise ratio. And here I provided two examples. So for example, on the left side, we have a CD20 protein staining. So guys, I need help. Who knows what is a CD20? Is there like anyone? Marker for which type of cells, if you know?
Something with the immune system.
Yeah, immune system. And if be more accurate?
Macrophage.
Almost. B cells. So it's a B cell marker on the left. And you can see that there are, so if you zoom in, you can see the rim of the particular cell. So for example, in this case, it might be easier. So we have the cell, and then we have the staining of the protein, which is like surrounds the cell. We don't see in this particular case the nuclei of the cell, but we just used the CD20. On the other side, it's like the same image in the same area. And if you look on the CD79A, which is also stains B cells, you can see that there is no signal for CD79A. I mean, like there are something like the speckles, but probably this is a noise. And in this case, the left image is nice because in this case we have like a signal. We can like distinguish noise from the signal. On the right side, it's not very well. In this case, probably this marker didn't work, so we don't see anything on the right side. And yeah, so this is also example how to generate image and add some noise computationally. And here just like to see that there is a clear signal without any noise. Then we have a high signal to noise ratio. So as you can see, there are, I hope that you can see in your kind of viewer that there is some noise on the background, but still we can like easily see the signal. And here we still can see the signal, but there is like a lot of noise on the background happening here. Yeah, so you can take a look on the recap, check it, and basically for today that's it. Next time we will go like further and we will explore some of the classical computer vision techniques, how we can, for example, reduce the noise, how we can detect edges on the image, and how we can threshold images. And basically, so the idea is that we will slowly go over the topics, and at some moment we will face with the segmentation task where we need like to take the image, something like that, for example, as a nuclei staining image, and we want like to draw the borders around the actual cells and measure different features inside of them. So this is our final task. So okay guys, that's it for today. If you have any questions, please, you are welcome to ask them. Or if not...
We have just 6 lectures according to the timetable.
Yeah, we will have just 6 lectures, I think. So basically it's supposed to be a short, kind of fast course, but in the end, the idea is that we will want to measure the features in our images and be able to analyze, clusterize, perform some of the typical single cell style techniques to analyze the image that we had in the beginning. So you already can go, for example, to HubMap resource to get the understanding of it. So on HubMap you can open the examples of the images. Do they have... So yeah, so here we have data types, data sets. And for example, if we open, let's say that we want to open lung. And then you can check whether they have... It's not very fast. Yeah, we want to check whether they have phenocycler. So they have phenocycler, which is a technique to get the spatial proteomics data. So I hope that it will work. Yeah, and let's just open one of the images. So yeah, and this is a visualization which is like on the HubMap. It's like some resource where people deposit their data for the public access. And here, this is a viewer of the data. So it takes time probably to load in the information that they have there. And the idea is that we want like to be able to do something similar. I mean, not to do spinners, but the actual results. Yeah, where is the result? Okay, let's try another way. So we can go to the visualization tool and check the examples. So yeah, so let's open something like that. So yeah, and this is an example of the actual data. So here we have a spatial viewer of the original image. Yeah, so it takes time to load because it's like in the web, and the probably application sometimes not the fastest one. But at the same time, for example, here you have the t-SNE plot of the same cells that viewer identified on the image. So here you can see that there are 2 channels right now toggled on, nuclei and poly-T. Yeah, I don't know why it's not working properly. But at the same time, you have the t-SNE plot of the cells that were defined there. And in case if it works, you can usually zoom in and check particular channels, proteins that were assigned to the cell, the expression of them, and be able to visualize them. So here, this is a panel of genes that are available. Yeah, but showcase didn't work, guys. Not worked as I was expecting. But the idea is basically, so yeah, we have the image. This is a input. The segmentation mask that you see on top of it, it's the result that we get after we applying the methods. Yeah. Okay, but this is the idea that we want to be able to do something similar.